Hello, my name is Ajramal Jamangarayeva. In my senior project, I studied how deep neural networks can predict mRNA subcellular localization under the supervision of Dr. Saipam. Let me share my screen. So our work was inspired by this publication in Bioinformatics Journal in 2019. Our initial task was to build a model with better performance than the tool that they developed. Unfortunately, we didn't have enough time to get better results, but however, we achieved similar results with the computationally less expensive model through changing the model architecture and using shorter input data. Why mRNA localization is important, and we have to study that. It is important because it plays a crucial role in protein production and regulation. Most of the cell's genetic material is contained in the nucleus. The mRNA is an RNA version of the gene that is produced in the nucleus, but it needs to get out into cytoplasm where it can be translated into protein. Cytoplasm is this liquid-filled space between nucleus and membrane. Therefore, mRNA localization will affect how many mRNAs are available for further translation and where this translation will happen. Real-life application of this research might include discovering defective trafficking in human cells, which is related to some muscular and neurodegenerative diseases as well as cancer. On the other side, understanding of mRNA trafficking mechanisms will improve the fundamental knowledge of molecular biology. For this project, we use CIFRA-seq data downloaded from GitHub. CIFRA-seq is a technology relying on biochemical separation of subcellular components. It was used to map gene transcript abundance in the nucleus and cytosol, as well as those associated to membrane. Membrane is this double layer of lipids and proteins that bounds the cell, and those left in insoluble fraction. Our goal is to build a deep neural network which can build mRNA localization values out of sequence alone. Let's say that we just input this long gene sequence into a model, and it should output this localization value. Let's review the first example to understand what we mean by localization value. So in the case of this first gene ID, this output means that 40% of the gene ID, the gene is located in cytosol. 16% of the gene is located in insoluble, almost 24% in membrane, and 90% in nucleus. Those numbers are localization values. Sometimes we refer as labels OY, and this model should predict it given only this gene sequence. To train our models, we collected data from two files. One is a FASTA file with gene sequences, and another is tabular file with localization values. After merging these two files, we got 11,373 gene sequences that we can use to train our data. This is only one example. This is example of one gene ID. Gene is a sequence, is a string of gene sequence is a string of 4 different nucleotides. One nucleotides, we have 4 different kind of nucleotides, A, C, G, and T. So sometimes I refer to nucleotide as a character. To build a prediction model, we use a machine learning algorithm. Machine learning maps the input data to a label data through matrix multiplication and backpropagation algorithm. Therefore, it's only possible to train prediction model with numerical data. We have to convert those gene sequences into multidimensional NumPy arrays. There are 2 steps necessary to implement this conversion. First, we have to decide the fixed length of this input data, and second, we have to convert those strings into numerical data. So we have more than 11,000 sequences. The shortest gene has 207 nucleotides, and the longest more than 34,000. To choose this fixed length, we came up with percentile tables, which means that 50% of all sequences are no longer than 3,300 characters. 90% of gene sequences maximum 7,300 characters, and 98% will have maximum 11,500 characters. To if let's say if we choose 4 kilobase, 4 kilobase means 4,000 nucleotides or 4,000 characters. If we decide to stick to this length, it means that all sequences that are shorter than 4,000 have to be padded with zeros, and all like sequences that are longer than 4,000, they have to be truncated. There are two ways to truncate sequences, either post or pre. Post means that we keep first 4,000 characters and we cut the right end. It's considering this, we decided to prepare 6 data versions to test. So 3 different input lengths, fixed lengths, and 2 different 4 kilobase truncated post, 4,000 truncated pre, and the same for other fixed lengths. And to turn our gene sequence into numerical data, we used one-hot encoding. One-hot encoding means that every nucleotide will be turned into a vector. Because we have 4 different nucleotides, every vector will have a length of 4. Like A will be represented as a vector 1, 0, 0, 0. C will be as vector 0, 1, 0, 0. In case of 8 kilobase, the final input data will be turned into this will be turned into this 3-dimensional matrix with 11,373 samples. Each sample will have a length of 8,000, and every character will be represented as a vector of 4. And so label data also needed some data preprocessing with Python pandas library. Originally, this file had more than 63,000 rows, but almost half of that had 0 localization values. But because FASTA file had only information for 11,000 sequences, we, after merging, we chose only those 11,373 samples. And originally, this TSV file had 2 localization values for each compartment, like for cytosol A, cytosol B, insoluble A, insoluble B. So first we found average values for each compartment, and then we computed those localization values as a percentage. Let's see, like that 40% in cytosol and 16% here. So to build the prediction model, we used convolutional neural networks, or CNN, sometimes we refer as CovNet, and recurrent neural networks, or RNN. CovNets perform particularly well on computer vision problems due to their ability to operate convolutionally. Those convolutional operations, they extract features from local input patches. Traditionally, 2D convolutions are used to detect 2D patches from image data. In the same way, we can use 1D convolutions extracting local 1D patches or subsequences from sequences. Such 1D convolutional layers can recognize local patterns in a sequence. Because the same transformation is performed on every patch, a pattern learned at a certain position in a sequence can later be recognized at a different position later. For instance, a 1D CovNet processing sequence of characters using a convolution window of size 5, like in this figure, should be able to learn words or word fragments of length 5 or less, and it should be able to recognize these words in any context in an input sequence. Also, additionally, 1D CovNets are light and computationally fast. But 1D CovNets process local patterns independently because they don't consider the order of those local fragments, unlike recurrent neural networks. It happens because CovNet looks for patterns anywhere in the input sequence and has no knowledge of temporal position of pattern it sees. If some combination of nucleotide is meaningful at some specific position of a gene sequence, the CovNet won't be able to produce the best results possible. Unlike CovNets, recurrent neural networks have memory and usually they are used in speech recognition and natural language processing. There are different kinds of RNN, for example, gated recurrent units or long short-term memory, or LSTM. Long or short-term memory networks are a type of recurrent neural networks with feedback connections. LSTM networks manage to keep contextual information of inputs by integrating a loop a loop that a loop that allows information to flow from one step to the next. LSTM networks manage this by learning when to remember and when to forget through the forget gate weights. It's like this forget gate, update gate, and output gate. They will track this information. In this project, we will try two different architectures. First, one consisting of only convolutional neural networks, and the second one with a combination of convolutional neural networks and LSTM. So this is a figure of the second model architecture. So first, like input a long sequence, 1D convolution will extract those local patterns, and as a result, we will have a sequence of those local shorter sequences like patterns. And then if that sequence matters, this recurrent neural network will see those like pattern in those sequences. All models were built using Python and Keras framework. Training of them, like data preprocessing was made on my local MacBook, but the training of the models was performed on the high performance computing cluster located in the business building of UHD. Here's an example of the first architecture with CovNets only. Traditionally, convolutional layers were successful in a pair with max pooling layers. So every convolutional layer in this project will be followed by max pooling. And we also used dropout with coefficient 0.2 to address overfitting issue. Like 0.2 means that 20% of weights will be randomly zeroed out. So this is an example with two convolutional layers. And finally, it's flattens the tensor into one-dimensional and attaches a dense layer with 4 output units with softmax activation function, which means that those 4 values will sum up into 1. So this is an example of second model architecture. So here we have 2 convolutional layers and 2 LSTM layers. Hyperparameter optimization is what makes the model accurate and applicable to real life. Parameters of the model are weights and bias. Hyperparameters are those that manipulate the way the weights and bias are computed. So examples of hyperparameters are number of iterations, epochs, number of convolutional layers or LSTM layers, number of filters in convolutional layers or number of outputs in LSTM layer, like kernel size in CovNet or like learning rate. There are many hyperparameters that could be tuned. So and I believe that there are 2 most important jobs of machine learning engineer. So the first is data preprocessing to prepare their input data and labels for their model, and second is hyperparameter optimization. As you saw, it's quite easy to build these models with Keras because thanks to built-in functions, but the main job is to find those perfect, like those hyperparameters that will result in good prediction accuracy. So but it would be unrealistic to try individual like kind of all permutations of those hyperparameters. So I try to automate this process by creating these 2 functions. One is create convolutional layer, which defines the model architecture. Like this for loop will set up there any number of convolutional layers in this model. And another is evaluate function, which compiles the model, trains the model, evaluates it on validation set, and does prediction, which is followed by the computation of Pearson correlation coefficients. And this nested loop will invoke those functions with parameter layer sizes. Layer sizes can have values such as like 32, which means that it will create 2 convolutional layers, each with 32 filters. If there are like 32, it means it will create 3 convolutional layers, each layer with 32 filters. So, and I also try to automate their reporting to make faster analysis. This code will create in some part of code here and here, this will create kind of table, which will include all results of this model. So after testing all these models on my 6 different data versions, I compared them in different categories. So here is the results. Here's a comparison between 4 kilobase, 8 kilobase, and 12 kilobase. So we can see that the best results were provided with 8 kilobase data and the worst with 4 kilobase. This is understandable because in the case of 4 kilobase, we had only 50%, only half of gene sequences with full length full sequence information. Another half were truncated, and in terms of 8 kilobase, 90% of gene sequences had full length information, and less than 10% were truncated. With 12 kilobase, only 2% of data was truncated. We notice no difference between 8 kilobase and 12 kilobase, but there is a difference between 4 kilobase and 8 kilobase, especially for compartments like membrane and nucleus. Those 2 compartments were hard to get good correlation coefficient, both in the publication and in our project. So because the best results were on 8 kilobase, we decided to stick on this data set. So here's a comparison between post-truncation and pre-truncation. As you see, we have best results for each fixed length when it's was truncated post than pre. Only in 12 kilobase there is no difference because only 2% of data was truncated, and it's understandable that there is no difference. But in 4 kilobase and 8 kilobase, post truncation resulted in better result. That means that gene information at the beginning at the left side has more importance than the gene on the right side. So then we compared 2 model architectures, like only convolutional layers and another combination of convolution and LSTM. We see that a combination gave better results, but the difference is not significant, which means that most of this correlation is because of the presence of these local patterns, which were extracted by convolutional layers. And sequence information is important, but it's not that much. The biggest value is in the presence of those local patterns. Here's a comparison between publication results and our project. As we worked on CFRES-Tech data, we can see this part, and we see that our RNA locator is a model with 5 convolutional layers and 1 LSTM, and this one is just 5 convolutional layers. As you see that our RNA locator results are very similar to the results of RNA tracker. RNA tracker was trained on full-length RNA inputs, which is much more computationally expensive, and the architecture, they had 2 convolutional layers, 1 LSTM and 1 attention layer. So we used callbacks in QRAS to automate epoch hyperparameter. As you can see, the best results were achieved at epoch 29, and we got the lowest mean absolute error and validation loss. So this is the structure of our RNA locator with 5 convolutional layers, 1 LSTM layer, and here so we have our visualization of Pearson correlation coefficients. RNA locator provided similar results as RNA tracker with simpler prediction model without the attention layer. Training with a fixed length input of 8 kilobase speeds up training significantly compared with full length inputs. For future work, it could be a good idea to test RNA locator with an attention layer. We could get much better results than the publication with fixed lengths because we increase the number of convolutional layers. And I think that if we add additional layer, we can even get better results. This project was a great opportunity for me to learn about deep learning. Even so, before I started on Coursera, like I got deep learning specialization course, I realized that only such a rigorous application of this technique can help to understand it. I learned better pandas library, NumPy structures, and matplotlib, and Keras. I'm very grateful. Thank you.